In 1984, Alec Jeffreys developed a technique for using DNA variation to establish human identity. He named his technique DNA fingerprinting. Jeffreys looks at a difference in DNA sequence called variable number of tandem repeats. It's also known as VNTRs. Difference in number of repeats, which one shown in orange boxes below, create different allele at locus on a chromosome. In this example, person A is heterozygous at this locus. He has different VNTR alleles on each of his pair chromosomes. Person B is homozygous. She has the same VNTR alleles on each chromosome. Jeffreys identified DNA sequences that match the VNTR repeats. Then he used this sequence as probe to detect additional VNTR regions. Jeffreys reasoned that when a restriction enzyme cut at a specific site on either side of the repeat region, the different number of repeats in the VNTR alleles would create restriction fragment of different size. A restriction digest of person's entire genome produce millions of DNA fragments of different sizes. Jeffrey realized that he could use his probe to identify the subset of restriction fragment containing the VNTR repeats. A restriction digest of a person's entire genome produces millions of DNA fragments of different size. Jeffrey realized that he could use his probe to identify the subset of restriction enzyme containing the VNTR repeats. The restriction fragments were located into an agarose gel and separated by electrophoresis. In this process, when electricity is applied, a small DNA fragment migrates faster through the gel matrix than longer one. Next, the DNA fragments were transferred to solid membrane in a process called southern blotting. To make the fingerprint, Jeffrey labeled his probe with radioactive and washed them over the membrane. The probes bound to any DNA fragment contain a VNTR. When Jeffrey exposed the membrane to X-ray film, radioactivity from the probe produced dark bands. This correspond to the positions of VNTRs. Jeffrey DNA fingerprinting method is known as multi-locus fingerprinting because his probe detects VNTR differences at multiple locations in genome. Although each person has unique multi-locus DNA fingerprint, the multiple bands are difficult to analyze. The result can be ambiguous if the DNA is not cut completely or when several alleles are located close together on the gel. Also, because many loci are analyzed at once, the frequency of one type or another in a population cannot be calculated precisely. This one is multiple bands, difficult to resolve. In 1987, a group of University Utah identified the VNTR locus named D1S80 on chromosome 1. This probe, which binds to unique sequence just outside the repeated structure, is termed as single locus probe. A restriction enzyme that cut at a specific site on either side of D1S80 locus creates fragments that correspond to different number of repeats in VNTR alleles. Unlike multi-locus probe, a single locus probe binds to only a single locations on a single chromosome. Thus, the D1S80 probe binds only to the D1S80 locus on chromosome 1 and nowhere else. The restriction fragments are loaded into gel and sized separated using electrophoresis. Then the millions of DNA restriction fragments are fixed in place on membrane. The radioactive probe is washed over the membrane. The probe binds only to allele to a specific locus in D1S80 locus on chromosome number 1. When the membrane is exposed to X-ray film, radioactivity from probe leave a dark band that corresponds to position of the D1S80 allele. Single locus fingerprint can be interpreted unambiguously and the frequency of different alleles can be determined precisely. So this is the multi-locus fingerprint on your left side and single locus fingerprint on your right side. This technique, multi-locus fingerprint, is defined by Alex Jeffrey. Another single locus fingerprint is the futuristics method which one use at a present time. Thank you.